We rely on one pediatric intervention to neutralize 3 fundamental bacterial threats. To understand the DPT matrix, we must analyze the specific pathology of each. The first is Corynebacterium diphtheriae. When this bacterium colonizes the upper respiratory tract, the primary damage is not caused by the bacterial cells themselves. As illustrated here, the bacteria release an exotoxin. This chemical byproduct triggers the formation of a thick, false membrane, physically obstructing the airway. This exotoxin also travels through the bloodstream to distant tissues, causing inflammation of the heart muscle and paralysis of the cranial nerves. The second threat, Clostridium tetani, persists in the environment as resilient spores in the soil, entering the body through puncture wounds or tissue damage. It produces a potent neurotoxin called tetanospasmin. This molecule binds to the myoneural junction of skeletal muscles and blocks the inhibitory pulses required for muscle relaxation, leading to rigid, agonizing contractions. With a case fatality rate between 10 and 20%, tetanus remains a critical risk for neonates, who are often infected through the umbilical stump in settings with inadequate aseptic care. Because diphtheria and tetanus kill via released toxins, the vaccine must be engineered to neutralize specific chemicals, rather than focusing solely on direct cellular destruction. The third pathogen, Bordetella pertussis, presents a different challenge. Unlike the exotoxin-heavy mechanisms of diphtheria and tetanus, pertussis is a direct mucosal infection of the respiratory tract. It is exceptionally contagious. In unvaccinated households, the attack rate for pertussis among close contacts exceeds 90%. This high transmissibility hits a vulnerable demographic. Up to 40% of all reported cases occur in infants under 6 months of age. For these infants, the infection often leads to post-tussive vomiting, cerebral hypoxia, and progressive encephalopathy. These complications result in a mortality rate of 0.5% for infants in their first 6 months of life. While diphtheria and tetanus are constant environmental risks, the immediate lethality of pertussis in newborns defines the precise start of the vaccination sequence. To counter the chemical threats of diphtheria and tetanus, scientists extract raw toxins and treat them with formaldehyde. This process creates toxoids, inactivated versions that cannot cause disease but retain the physical structure needed to train the immune system. These toxoids are then combined with whole-cell Bordetella pertussis bacteria and adsorbed onto aluminum salts. In this formulation, the whole-cell pertussis component serves 2 roles. It immunizes against whooping cough, and it functions as a powerful adjuvant. The reactive nature of the whole-cell bacteria aggressively stimulates the immune system. This adjuvant effect forces a more robust response to the diphtheria and tetanus toxoids than they would generate on their own. The traditional DPT shot is a calculated compromise. It utilizes the inflammatory properties of whole-cell pertussis to ensure the body mounts a defensive response against 3 different diseases at once. Establishing clinical immunity for diphtheria and tetanus requires a circulating antitoxin level of 0.01 international units per milliliter. Achieving this level depends on timing. Infants begin with high maternal antibodies that dissipate over months. If administered while these are high, they suppress the infant's own immune response, leaving them with low long-term protection against diphtheria and tetanus. However, pertussis is the exception. Maternal antibodies provide virtually no protection against severe whooping cough. Pediatricians must weigh the risk of a suppressed diphtheria and tetanus response against the immediate high-mortality threat of pertussis. To maximize survival, the schedule is accelerated, with the first dose given at 6 weeks of age. This strategy prioritizes immediate pertussis protection, accepting a lower initial response to the diphtheria and tetanus toxoids that will be corrected through subsequent booster doses. While effective as an adjuvant, the whole-cell pertussis vaccine, or DTWP, contains multiple raw endotoxins, leading to high rates of adverse reactions. These reactions include high fever, inconsolable crying for several hours, and hypotonic hyporesponsive episodes, where the infant becomes briefly limp and unresponsive. Researchers developed acellular pertussis vaccines, known as DTaP, as a safer alternative. These contain only specific detoxified proteins from the bacteria, such as pertactin and filamentous hemagglutinin. Large-scale randomized controlled trials in Sweden and Italy, involving over 100,000 children, were conducted to validate this new formulation. Results showed high efficacy. Acellular vaccines protected approximately 85% of recipients from pertussis. Traditional whole-cell vaccines performed significantly worse, reaching only 36 to 48% efficacy, while maintaining a much higher rate of systemic side effects. The acellular formulation represents a shift in vaccine design. By isolating specific antigens, it provides a superior safety profile while matching or exceeding the protection offered by the whole-cell standard. At the national level, consistent vaccination programs have successfully eliminated diphtheria in many regions. Pertussis remains a challenge. While the vaccine prevents severe disease, it does not completely halt the colonization of the bacteria or its transmission to others. Since immunity from both the vaccine and measles wanes over time, maintaining the 0.01 international unit threshold requires a rigid series of boosters at 12 to 24 months and again at 4 to 6 years. Because these pathogens cannot be eradicated, continuous population-level vaccination and strict adherence to the booster schedule are permanent requirements for public health. While the clinical data is clear, health economics presents a final barrier to the universal adoption of the acellular vaccine. In Malaysia, a single dose of the traditional whole-cell DTWP vaccine costs approximately 0.78 ringgit. The bioengineered acellular DTaP formulation costs 28 ringgit per dose. This 35-fold increase in cost creates a massive budgetary challenge when applied to an entire national birth cohort. The clinical evidence mandates the accelerated 6-week schedule and the superior safety of the acellular formulation. Transitioning the global population to this standard now depends on reconciling the extreme cost differential of the vaccine.